Okay, hi guys. I'm hoping, yeah. Okay, I will just turn off camera because I'm not in the best mood. But anyway, so this is the lecture that I didn't finish on October 24th. So this lecture will actually come up for our quiz 3, and of course it will be on final. It's not hard lecture, and we heard most of these things during our class. So thinking about viruses, so what viruses are? So, and this will be a question in your quiz. And it's important to know that viruses have no kingdom, so they're not members of any of the kingdoms. They're not plants, they're not animals, but we also say that they're not bacteria or protists. So they're kind of very special, not even an organism, they're very special structure is how I like to say. So we define viruses as infection agents, and they're made of nucleic acids. Nucleic acids are DNA or RNA, and usually they're coated in a protein coat that is called capsid. Viruses are obligate cellular parasites, which means that they can live only inside of a host cell, but you will see later in this lecture, and be very careful about that during the quiz and the exam, because viruses, even though they cannot live out of cell, they can survive out of cell for some time, and that varies between different species. Hosts can be bacteria, animals, or plants, and different viruses attack different organisms. And we say that viruses are smallest of all microbes. Basically, to cover the head of a pin, it will take 500 million of viruses. So they're pretty teeny tiny structures. They come in many different shapes and sizes, and usually we say that even though they have such a different shapes and different sizes, of course, there are two main essential components that will make each of these very different viruses. One of them is genetic component, genetic material that comes in the form of DNA or RNA, and viruses can have single-stranded DNA and double-stranded DNA and single-stranded RNA and double-stranded RNA. So they're quite diverse. So that's the first component. Second component is protective protein coat, and that coat is called capsid. So that's their protein structure, and that structure surrounds genetic material. And this picture here shows you only how diverse viruses are, and they look kind of nice and cute. My favorite is bacteriophage, this one here. So in terms of characteristics, what we say is that viruses have no organelles or nucleus, they have no cytoplasm or cell membrane, and they are non-cellular or acellular. So that means they're not members of any kingdom. So the way how they exist is by extracting the cellular machinery to another living thing to reproduce by injecting its genetic material into a host cell. So in other words, they're trying to deliver their own genetic material to the host cell, and then they are using host cellular machinery to reproduce. So viruses can be either with envelope or non-enveloped or naked. There are different structures. So some of them may have outer envelope, and if they have outer envelope, that envelope is a membrane that originates from host cell. Now that envelope, again, envelope originates from the host cell, and very often has specific spikes, like on virus influenza, which is a flu virus, has those H and N spikes, and those spikes actually help in attachment and very often makes them sensitive to chemical actions of disinfectants. Now, in terms of viral evolution, there are three main hypotheses how virus arose and how they developed, and we will discuss briefly all three of them. Again, this year I didn't have a chance to get the professor who is an expert in virus evolution, so he would definitely be better than myself to tell you about viruses. But for now, what you need to know is that there are three hypotheses, and the first one is virus early hypothesis, which clearly says that at the beginning there were replicative elements. So those replicative elements preceded very early life forms, and then viruses evolved from this ancestor. So that pretty much means that viruses were first. Second one is a regression hypothesis, which tells about degeneration of cell was the basis for viral rise. So later on, those degenerative cells acquired fully parasitic lifestyle. So that basically means going in reverse. So first cell developed, and then it lost a bunch of characteristics, and then virus developed from that. So basically going in reverse. And the third one, which is called escaped gene hypothesis, says that cellular genes, so the first cell simply arose as we talked about, but then cellular genes simply acquire the ability for self-replication and spreading. So again, in my opinion, even though I'm not virus expert, third hypothesis makes more sense. But as I said, this is just my educated guess, and it makes sense to me, especially because RNA has self-replicative capabilities, and we know now theory of RNA world is now established. So to me, this makes total sense. But again, not the best to talk about that. In terms of structure, viruses have variable size, so from 10 to 400 nanometers, each of them very small, and most of them can be seen in electronic microscope. All virions contain nucleocapsid, which is composed of nucleic acid, DNA, or RNA, and protein coat, and protein coat is called capsid. Again, they all must have DNA or RNA or protein coat, which is capsid. Some of them have additional envelope, some of them don't have envelope, but that envelope originates from host cell. So this envelope, again, is flexible membranous layer, and animal viruses envelope originates from host plasma membrane or nuclear membrane. And the envelope is kind of important as a viral ID, so it's used to identify the type of viruses, and many of those envelopes have enzymatic activity, like in influenza virus. This enzyme neuraminidase is very famous portion of envelope. Sometimes envelope can play a role in nucleic acid replication, but again, it's important to note that not all viruses have to have envelopes. But if they have, it's quite important as their viral ID. So on viral envelopes, as we said, we can see many enzymes. And it was first thought that all virions actually lack enzymes. Now it's accepted that a variety of virions actually have enzymes, like reverse transcriptase. So enzymes are not only present on envelopes, they can also be inside of the virus. And this reverse transcriptase is a great idea, is a great example. So what you need to understand, what I'm trying to say with this, is that viral enzymes can be located on envelopes, but they can also be inside of the mature virion. Certain viruses can attack only certain cell types, and this actually functions like a key lock system. So it's like making a pieces of puzzle. So the ends of one piece of puzzle must match up, so the ends of one piece of puzzle have to match with another piece of puzzle. So as you can see, surface marker and receptor site. So that means that viral antigen will only specifically bind to the receptors on the T cell, and this functions as a key lock system. So literally, virus recognize cell, and it can infect it by just matching the surface marker with the receptor site on a cell. So again, the example, rabies virus only attacks brain or nervous cell because of specific surface receptors. So viruses have big variation of size, but they are still smaller than bacteria. There are some big viruses that are close in size to E. coli, but there are also some teeny tiny small viruses, and they're quite variable. You can see that parvovirus is like 20 nanometers, and some variola viruses is 360. So again, highly diverse. Ebola is huge. Ebola is really big. In terms of classification of viruses, we will touch up a little bit of morphology and replication. So morphology of viruses when we say morphology of viruses, we refer to the symmetry of viruses. And viruses have different types of symmetry. They're helical, icosahedral, or complex symmetry. I am pretty much not, I'm not very interested in details about recognizing these types of symmetry, but what I want you to know is that viruses have different types of symmetry and they're quite diverse. Helical symmetries, polyhedral viruses, spherical or complex viruses. Again, for now, I do want you to know that bacteriophage has a complex symmetry, simply because bacteriophages are viruses that infect specifically bacteria. And it might not be bad for exam to know that coronavirus belongs to the spherical viruses, simply because we are so affected with coronaviruses pandemic in past few years that it would be really nice if we memorize that it's actually spherical virus. And again, this is just a picture showing you how they look. So this the top picture is the drawing or cartoon, and the bottom picture is their electron micrography. And again, my favorite is bacteriophage. It is really amazing organism, and that's why I will talk a little bit about bacteriophages. But first, replication of viruses. So viruses have RNA. Viruses can have RNA or DNA gene. Most of viruses are RNA viruses, and basically what that means is that inside of the capsid it can have either single-strand or double-strand RNA virus. So this is very unique. So viruses are unique there because they are only organisms that have double-stranded RNA. And if they are DNA-based viruses, that means that they have linear double-strand DNA in genome, and some exceptional case contains circular DNA genome and single-strand DNA. So there are examples of single-strand DNA and circular DNA viruses. And the virus replication cycle will be more important for the final exam than for the quiz. I can definitely tell you because I already make questions for the quiz. But for the final exam, this will be very important, especially the order of how virus replicates. So the step number one is attachment. The attachment means that the virion attaches to the host cell, basically key lock system, so recognizing a specific surface receptor on the host cell. The next step is penetration, or other words for penetration is viral entry, and that's the moment that comes after attachment when the virus or viral nucleic acid gains the entrance into the cell. Now, when virus is in cell, the third step is synthesis. What that means is that the viral nucleic acid and protein copies are manufactured or synthesized by cell's machinery. And this is important to know. So viral DNA is actually exploiting cellular machinery to synthesize their own components. The fourth step is assembly, when virus is produced from viral components. So cell already synthesized all capsid components, replicated viral nucleic acid. And the next step is release, which is the moment when new formed virions are released from the host cell. So for the virus attachment, it's important to have specific receptor, and those receptors can be different between naked and viruses with envelope, but in fact, it functions with the principal key lock system. In terms of viral entry system, there are three methods that are used by virus to enter, and that's fusion, endocytosis, or injection of nucleic acid. Fusion is a method of entry which assumes that basically the viral envelope is fusing with a host membrane, and then nucleocapsid all enters. Endocytosis in vesicle is the type of penetration where we actually have endosome that helps in viral uncoating, and then there is a penetration. Or the most simple is injection of nucleic acid, so just like a needle. Then third step is synthesis, when the viral nucleic acid and protein copies are made by cellular machinery. So again, virus cannot reproduce alone. It must come into the host cell, and then eventually it's exploiting its resources and lyses it and killing it. Uh, when virus enter the cell, it makes copies of itself and causes cells to burst to make more viruses. And again, if we talk about, and we will see that when we talk about bacteriophages, if cell is lysed after releasing of virus, we talk about lytic infection, or if it's not lysed, if virus persists just being a dormant in cell, we talk about lysogenic infection. And this is the best example for this is bacteriophage. And in lytic infection, virus will simply enter the host cell, makes a copy of itself, and cause cell to burst or lyse. In lysogenic infection, virus doesn't let the host cell to lyse away. It remains dormant in cell, and usually viral nucleic acid gets integrated in the host DNA and then stays there dormant. It doesn't do anything. If there is some sort of stressful condition, that can cause switching from lysogenic to lytic cycle, and then cell will get lysed and burst. So how long can virus survive out of the host cell? So surprisingly long, but that really depends on the environmental conditions like a moisture or temperature. So they live longer on water-resistant surfaces like a stainless steel or plastic, and the cold virus sometimes can survive on indoor surfaces for several days. So, but it won't be so infective after it was outside for a while. Flu viruses apparently can survive in the air for several hours, and apparently they survive better at lower temperature, while enteric viruses can survive for weeks if conditions are suitable. So they're not so meaningless out of living cell, but they cannot reproduce. So virus in the organism is disrupting our normal balance, and we can prevent virus with vaccines, but not with antibiotics. Antibiotics kill bacteria, and this is something that I'm facing with pretty much my whole life. When you have a flu or something that is apparently viral, the doctor will just, oh, take some antibiotics. And I'm like, no. Anyway, so positive aspects of virus is that you can apply them through genetic engineering. So harmless viruses carry good genes into the cell because they have genes that will help entrance in the cell. So it's very common to have viral vectors that will help in transfection when you're trying to modify a eukaryotic cell. And there is also phage therapy when bacteriophage, along with antibiotic, is used against antibiotic-resistant bacteria. So it's and also bacteriophage phage displays very commonly used to produce antibodies these days. Again, if somebody is interested, I can talk more about that during office hours. And this is just basically the list of viruses that I'm trying to give you a sense of what is there, but please don't memorize all of these viruses. That's not what I will ever ask you to do. Plant viruses is tobacco mosaic virus that infects crops. Animal viruses are bird flu or that was very big deal a few years ago. Human viruses, of course, COVID, common cold, but please don't memorize all these viruses. That's not what I will ever ask. And also this table. So this is just for you to show you what are basic differences between bacteria and viruses. Again, this is not something that I will ever ask in details, but it is important to know that bacteria are living organisms and viruses are not considered, they are considered as non-living and are living organisms only inside of the host body. So they're kind of on the border. Bacteria are free living, so they can live even outside of the body. Viruses cannot. Bacteria are larger, 2 to 8 micrometers. Viruses are much smaller. So don't memorize numbers, just know that bacteria are larger. In terms of structure, bacteria are unicellular and have single circular chromosome. viruses do not have cell. They only have nucleic acid with a protein coat instead of cell wall. So viruses do not have cell membrane or cell wall, and bacteria are reproducing by themselves with binary fission. Virus only grows and reproduces in the host cell. Bacteria develop localized infection. Viruses cause systemic infection, and just some examples of bacterial diseases: pneumonia or tuberculosis, while a viral infection is AIDS or COVID. Benefits of bacteria are that they can be beneficial for human body in many cases, especially in microbiome. Viruses, on the other hand, can be very harmful, but they can be beneficial for us in terms of genetic engineering. And prevention of bacterial infection is with antibiotics, while viruses are prevented with vaccines. In terms of bacteriophage, also known as a phage, this is something that I do like to ask, and that's a virus that replicates in bacterium, attaches itself and infects the host cell, and host cell is bacterium. Bacteriophage has enzymes that destroy bacterial cell wall from outside and inside, and they do that by hydrolyzing carbohydrate and protein components. And some of the examples of bacteriophages are bacteriophage T4 and T1 to T7. So T is the type. So very famous viruses that you can see here. This is how this is the picture of bacteriophage. And bacteriophages are known by extensive gene exchange and the absence of homology at nucleotide level between phages. So they have their small number of different variants. What I'm trying to say is that they're quite diverse, and it's hard to get homology between them. So the question is, are those structural similarities in phages, and they are structurally similar, most of them look like that, are they a result of divergent or convergent evolution? So this is what I'm going to ask on the quiz. So how did viruses, or specifically bacteriophages, evolve? So either divergent or convergent. So what means divergent? So if they evolved by divergent evolution, that means that they all share common ancestor and they maintain basic architecture of structural proteins, but then they diverge so much that you cannot see their homology. If it was convergent, that means they do not share common ancestor. They simply converge toward the structure that was optimal to build or infections particle. So which one is correct? The main difference that you have to make here is whether they whether they share common ancestor or not. If they evolved by divergent evolution, that means they had common ancestor and then they simply developed and they evolved so much that homology is not possible to be detected, or they just don't share common ancestor, they just converge toward the structure that was optimal forever. So both types of evolution led to the single common trait that helped them to survive in host cell. But apparently, based on the accumulation of structural characteristics, it's likely that there was a divergent evolution. That means they had common ancestor. So what are evidences of divergent evolution? Well, similarity between domains, of course. So this, again, don't memorize, please, the names of these viruses, but this specific phage capsid, this specific phage had the major capsid protein MCP that was highly similar to the archaeal virus Steve 134 and mammalian adenoviruses. So basically, the point is that there was similarity between bacterial, archaeal, and mammalian viruses, and the relationship was found between tailed double-stranded DNA phages and archaeal virus. So, and similarity of phages 5, 6, and 5, 8 with eukaryotic viruses. So, long story short is that all similarities that they found that exist between all three domains, between viruses that infect all three domains of life, are an evidence that viruses actually evolved by divergent evolution. And there was this great paper that I can share with you if you are interested. And this is basically it talks about how viruses are important for evolution. So basically, what they are talking here about is a viral shunt in soil. So it's published in 2023. So what they did here is they isolated soil microbial community, and then they performed the analysis of soil microbial community with viruses, and they noticed that there is then they examined nutrient and dynamics of microbes in soil after viral lysis. The way how they do that, they check dissolved organic matter in the soil before and after viral lysis. So their question was, does viral lysis of bacterial cells in soil affect soil nutrient characteristics? And the answer was yes. So at two different temperatures, nutrient limitation was removed after viral lysis. So what was their conclusion is that viruses actually increased dissolved organic matter in soil, and then microbes survived because they reutilized that dissolved organic matter that was released by viruses. So the way how they saw increased metabolic activity of soil after viral lysis. So that's basically viral shunt in soils. And viruses are important for the evolution, especially because RNA, then they have RNA in their genome. RNA is the only molecule that can do reverse transcription from DNA, and it exists in RNA viruses. And this enzyme, RNA polymerase, RDRP, is the only universally conserved hallmark protein in RNA viruses. And this enzyme, reverse transcriptase, is actually key for reconstruction of viral evolution history. So if you remember, we talked about how RNA was first and then DNA took over as a more stable molecule. But guess what? The only enzyme that can actually make DNA from RNA is reverse transcriptase. So again, this is typical enzyme for viruses. There are no homologs with same enzymatic activities. And other organisms. So that's why this enzyme is quite important for evolution. And that will be all from me this time. See you tomorrow.